Bismillahirrahmanirrahim. Assalamu alaikum. This is Amanullah. Today you're watching my YouTube channel, Dr. Aman's video. Dear viewer, today we will learn about the deoxyribonucleic acid or DNA. That we know that DNA carry necessary information for the life, or DNA carry information from one generation to another generation. Like for example, in human being, it is the DNA which carry information for the color of the hair, for the color of the eyes, for the height, for the skin color, and for any other genetic disorder if there is present. So it is the DNA which carry information for the life. DNA is present in nearly in all living cell, in eukaryotic cell, in prokaryotic cell, and also some viruses which contain DNA. Because there are certain other viruses which do not contain DNA, they are RNA containing viruses. So now the question is, what is DNA? DNA is a chemical compound which is made up of a basic structural or functional unit which is known as nucleotide. Nucleotide is itself made up of three components. Number one is deoxyribose, number two phosphate group, and number three nitrogenous base. So nucleotide is made up of these three components. Now we will discuss that what are these three components. First of all, deoxyribose sugar. Deoxyribose sugar is actually a monosaccharide carbohydrate molecule which is composed of 5 carbon. This is, in this diagram you can see that this is the deoxyribose sugar which contain 5 carbon. This is the carbon number 1, this is the carbon number 2, this is the carbon number 3, 4 and 5. So this sugar is a deoxyribose sugar which is pentose in nature. It means that it contains 5 carbon, therefore it is pentose in nature. Now, why it is called deoxyribose? It is called deoxyribose because the original ribose sugar contain hydroxyl group at carbon number 1, 2, 3, 4 and 5. But this deoxyribose molecule do not contain hydroxyl group at carbon number 2. Therefore, it is 2-deoxyribose sugar. It means that this ribose molecule did not contain a hydroxyl group at carbon number 2. Therefore, it is a deoxyribose sugar. So, one component of nucleotide is deoxyribose sugar. The second component is phosphate group. So phosphate group is always attached to the carbon number 5 of the deoxyribose sugar. Here you can see this is carbon 1, 2, 3, 4 and this is 5, and phosphate group, this green P indicate phosphate group. So the phosphate group is attached to the carbon number 5. Now, the third member or the third component of the nucleotide is nitrogenous base. What is nitrogenous base? We will discuss little later. But the nitrogenous base is always attached to the carbon number 1 of the ribose sugar. Here you can see this black box actually indicate a nitrogenous base which is attached at the carbon number 1. Now this complete molecule, nitrogenous base, pentose sugar or deoxyribose sugar with phosphate group, is known as nucleotide, and this nucleotide is the basic structural and functional unit of the DNA. But DNA is a polynucleotide structure. It is not composed of a one nucleotide, it is a polynucleotide structure. So therefore one nucleotide form bond with another nucleotide and then so on and so on. In this way they form a polynucleotide structure. So how one nucleotide attach with another nucleotide? So if we say that this is the nucleotide number 1, this is 2 and this is 3, then here you can see that this 3' carbon or 3 carbon contain hydroxyl group, and this 5 carbon contain phosphate group, because at carbon number 5 there is always a phosphate group. So this phosphate group form a bond with this hydroxyl group in the carbon number 3 and produced a bond which is known as phosphodiester bond. Phospho, it may not be able to see to you, phosphodiester bond. So the phosphate group present at the carbon number 5 and the hydroxyl group present at the carbon number 3 of another nucleotide. This is nucleotide number 2 and this is nucleotide number 1. So these two produce a bond which is known as phosphodiester bond, and this phosphodiester bond linked one nucleotide with another nucleotide. And now this second nucleotide produced another bond between hydroxyl group at 3' carbon and phosphate group at 5' carbon. So another phosphodiester bond is formed. So these 3 nucleotides are linked with each other through a phosphodiester bond between hydroxyl group at carbon number 3 of one nucleotide and phosphate group at carbon number 5 of adjacent nucleotide. So in this way they form a polynucleotide structure. Now this is nucleotide number 1, this is nucleotide number 2, this is the nucleotide number 3. So it is a polynucleotide structure. This polynucleotide structures formed a single strand of DNA. As I told you that DNA is a polynucleotide structure. So now this is a polynucleotide. This polynucleotide is actually a single strand of DNA or one strand of DNA. Now it is very important that at the end of last nucleotide there will be a free hydroxyl group for further bonding. And we know that this hydroxyl group is always present at 3' carbon. So this end will be known as 3' end of DNA. Again I repeat that at the end of the at the last nucleotide there will be a free hydroxyl group for further bonding at carbon number 3. Therefore this end of the DNA strand is known as 3' end. And you can see that at the upper end there will be a free phosphate group at carbon number 5 for further bonding. So therefore this end is known as 5' end, and the orientation of this DNA strand is 3' to 5'. Now this is the one strand of DNA. Now we know that DNA is present in the form of double strand, that there are always two stranded which are attached to each other, and the structure of these two strandeds are known as double stranded DNA. So now we will discuss the another strands of the DNA. Here you can see that the orientation, this could be the right position. You can see this is also the same polynucleotide structure as we have made here in this strand, but the only difference is the orientation of deoxyribose molecule is opposite to these molecule. You can see the orientation of this molecule is upright while the orientation of this molecule is down, towards down. So therefore the orientation of one strand will be always opposite to the other strand. Why we need this opposite orientation, we will discuss in the coming words. So here you can see that in this strand there is a free hydroxyl group present at the carbon number 3rd, because the orientation is here different. This is carbon number 1, this is 2, this is 3, this is 4 and this is 5. So at the upper end there will be a free hydroxyl group at 3 prime carbon for further bonding, and at the lower end there will be a free phosphate group at carbon number 5 for further bonding. There will be a phosphodiester bond between the 3 prime hydroxyl group and 5 prime phosphate group. So there will be a phosphodiester bond and there will be another phosphodiester bond, and these 5 phosphodiester bond will attach these nucleotide with each other. But there will be a 3 prime hydroxyl group which is free for further bonding, and there will be a phosphate group which is at 5 carbon number 5 for further bonding. Therefore, the orientation of this strand will be 3 prime to 5 prime, and the orientation of this strand is 3 prime to 5 prime. So it runs from 3 prime to 5 prime, and it runs from upward from 3 prime towards 5 prime. Is it right? So now it is one single strand and this is another single strand. Now how these two strands attached with each other? So it is very important, as we know that at carbon number 1 there is a nitrogenous base attached in the nucleotide, and here you can see there is another nitrogenous base. So these nitrogenous bases will form bond with each other. Like for example, so before going into the bonding, I would like to tell you that we know that there are total 4 nitrogenous bases which formed DNA. One is adenine, another is guanine, third one is cytosine, and fourth one is the thymine. So these are the 4 basic nitrogenous bases which formed structure of DNA. The adenine and guanine are purine bases. Again I repeat, the adenine and guanine are purine bases. So purine bases are made up of 2 rings. I have made here, but it is very small structure. If you can see, these are the 2 rings. One is 6-member ring and the other is 5-member ring. So this 6-member ring and 5-member rings fused with each other and they form a structure of purine bases, which is adenine and guanine. While the pyrimidine bases are cytosine and thymine, and the structure of pyrimidine bases is composed of a 6-member ring. Here you can see that it is a single ring which is 6-member or which has 6 corners. Here you can see it is a 6-member ring. So the purine always bind with pyrimidine. It is not possible that purine will bind with purine or pyrimidine will bind with pyrimidine. No, it is always a combination that purine base bind with pyrimidine. But here it is more specific that the purine base adenine will always form bond with thiamine. Again I repeat, adenine, which is purine in nature, will always form bond with thymine, which is pyrimidine in nature. So guanine, which is again a purine base, always form a bond with cytosine, which is pyrimidine base. But here you must know that adenine always form 2 hydrogen bond with thiamine. The bond between 2 bases are always hydrogen bond, and we know that hydrogen bond is the bond between hydrogen and the most electronegative atom. So adenine always form 2 hydrogen bond with thymine and it is always specific, and guanine always form 3 bond with cytosine. So the bonding pattern is always specific. Adenine will always form bond with thymine and guanine will always form bond with cytosine, and this specific bonding pattern is known as complementary base pairing. That the pairing of the bases with each other is always complementary. It is specific. It will not form bond without its contemporary base pair. Now, the comes to the opposite orientation, as I told you that the orientation of the single strand will be opposite, and I have asked you that why do we need this opposite orientation. So we need this opposite orientation for this complementary base pairing. If this orientation is not opposite, then these bases are not able to form complementary bonding with each other. So therefore, the orientation of two strands will always be opposite to each other in order to facilitate the pairing between bases or the formation of hydrogen bonding between bases. Now, if you can see carefully, it looks like a ladder, you know, ladder. The basic structure of the DNA is look like a ladder. It is the one side of the ladder, it is the another side of the ladder, and the bases form rungs of the ladder, rungs of the ladder. So the basic structures of the DNA is look like a ladder. It will like this. It is one single strand, it is another single strand, 3' to 5', 3' to 5', it is opposite direction. Sorry, I think you are not able to see these lines, so I may draw with another pen. Now I think it is more visible. Yes, 5', 3', and opposite direction 5' to 3'. Now this is the one strand, this is the another strand. Now these are the complementary bonding between bases. So it is the basic structures of the ladder. So the basic structure of the DNA is look like a ladder. But we know that the thread of the DNA in a single cell is very long. So therefore, the DNA twisted around itself. The DNA twisted around itself and form a helix or a spiral structure. Form a helical or a spiral structure, and therefore the three-dimensional structure of the DNA is double helical structure or double helix. So the three-dimensional structure of the DNA is double helix. So it twists around itself, or you can say the DNA coil around itself or around a special protein in order to make a peak of itself, in order to accommodate itself inside the cell, because otherwise the long thread of the DNA will not be able to accommodate itself inside the cell. So this packing, so this the pre-packing or the coiling of DNA is known as packing of the DNA. So DNA pack itself in order to adjust inside the cell, because otherwise the long threads are not able to adjust itself inside the cell. So it was all about the very basic structures of the DNA. I hope you have enjoyed this lecture. If you have got any information from this lecture, then I would of course request to subscribe my YouTube channel Dr. Aman's video, and try to hit bell icon in order to get notification for my upcoming video. Thank you so much for watching my YouTube channel. Fi Amanillah.